In today's lesson, we'll introduce a laser-based technique called flow cytometry. Flow cytometry detects and measures specific cells or molecules based on their physical and chemical properties, such as fluorescence, size, and complexity. Flow cytometry can also be used to analyze the cell cycle and cell viability. Because of its ability to characterize and count the number of cells, flow cytometry can help us diagnose and classify diseases, such as leukemia. When a sample containing the cell is introduced into the cytometer, it passes through a nozzle. The nozzle contains a piezoelectric device, which produces acoustic waves with a frequency of 2 MHz or above. Through these waves, the aggregated cells and air bubbles are removed. The inside of the nozzle contains the sample, while the outside acts as a reservoir for the buffer. These two components help the hydrodynamic focusing, a phenomenon in which fluids surrounding our sample allow cells to flow in one line. When a sample flows from one cell at a time, it passes through a laser beam and is further categorized by the detector, a photomultiplier tube. The first type of detector is the front scatter detector, which is parallel to the laser beam. The second type of detector is a side scatter detector, located perpendicular to the laser beam. The forward scattering occurs because of the diffraction of light passing from the sample, providing information about the cell size. At the same time, the side scatter detects the granularity of cell complexity of cells and fluorescence. Simply, cells with low granules will show more forward scattering, while cells with high granules will show high side scattering. Cells will also be separated based on their difference in fluorescence. When the cell emits fluorescence, the emission filters, detects, and records the fluorophores in a computer. Then, the computer converts the data into a histogram plot, making the x-axis the forward scatter and the number of cells for the y-axis. Like the forward scatter, the side scatter light is converted to a voltage pulse, which is directly proportional to the amount of side scattered light. Lastly, cells can also be separated by their physical properties. As mentioned, the hydrodynamic focusing allows our sample to flow in one line. With some distance, the line flows into tiny droplets containing a single cell. Then, the tiny droplets pass through electrically charged plates, causing the droplets to deflect by the electric field and separate into different containers. To summarize, the flow cytometer allows us to measure our sample cells' size, fluorescence, and complexity, providing us with an efficient yet accurate instrument.